how do we actually assemble our DNA, our insert, into our plasmid vector? Well, the traditional canonical way is with restriction, digestion, and ligation. So this is a very simple strategy. You have your plasmid, which contains an MCS, which contains various restriction enzymes. For the purposes of this video, we'll pick EcoR1. So EcoR1 has the restriction site G, A, T, T, C. And it's a type 2 restriction enzyme, which means it forms a dimer, cuts. It forms a dimer with itself, so it cuts palindromic sequences. So the other sequence is C, T, T, A, G. And if you can't see what I mean by palindrome, G, A, T, T, C, and reverse complement is the same, G, A, T, T, C. EcoR1 actually cuts there. So it leaves the G and it cuts everything else off. Now, this is very useful for opening up our plasmid. Because if I redraw this with the open plasmid after EcoR1 has been applied, and I make this a bit bigger so that I can actually draw with it, then on this side, we will have C, yes, we will have C, T, T, A, and then there will be a G there. And on the other side, we will have G up top, and then on the bottom, it will be C, A, T, T, C. And if I draw this with this L shape, as I did before, and on this side, you can see that those two can religate because this restriction enzyme has left sticky ends that are complementary to each other. Now, from now on, I'm going to not draw out the entire lettered sequence. I'm just going to represent it with these L shapes. So on the left-hand side, on the 5' end, we have that L shape, and on the other side, we have the complementary one. Now this can religate, but it's also very exploitable. We can use this. If we create an insert that looks similar, so here's my insert, and on one side of my insert, I'll have a restriction site like so, and on the other, I'll have another one. So this is all EcoR1 restriction sites. And then I'll have spacers, which are for nucleotide sequences, usually GATC, that just let the restriction enzyme bind better. And when you introduce EcoR1 into this reaction mixture, it will cut these so that it leaves complementary sticky ends, like so. And what you will probably see is that now our insert can ligate into our plasmid. And I'll show you what that looks like. Through my pen. Okay, so insert sits there and goes across. And the other side, ligate, is complementary there. So that's our insert set to our plasmid. However, there are some issues with this. This is incredibly simple, incredibly easy to do, but there are issues. So for one, restriction digest and ligation, unlike other techniques, is not orientation specific. So if I start replacing these with colors, these are all EcoR1 restriction sites. So they're still the same, they're just different colors for illustrative purposes. If this is our plasmid, then if I draw my insert, well, the orientation I would want after it's been digested will be red on red, blue on blue. So there's my insert, and there's the second restriction site. You can see how that will ligate. Red will go to red, blue will go to blue. But I can rotate that insert around 180 degrees. And what I'll get when I do that is on this side, it will be blue. And then I'll have my insert. And then on the other side, it will be red. And when that ligates, when this second one ligates, it looks like this. And you might say, oh, well, red and blue is the same, so it's fine. But the issue we have is that this insert, if I label it with dashes, let's say that's 1 dash, 2 dash, 3 dash, then what I want on this insert is for it to be in that orientation. Because maybe it's a coding sequence that needs to be read that way. Maybe it's a promoter that needs to be read that way, right? But when I rotate it through, it looks like this, where those are the wrong way around. And if it's that incorrect orientation that religates, then I do get that incorrect order, that incorrect orientation of my insert. So that's one issue with restriction enzyme and religation, is that you don't always get the correct orientation. So you have to do additional screening of your transformed colonies, usually with orientation-specific screening plans. That's not the only issue. The second issue, if I go back to red for everything, is that, as I mentioned, the plasmid backbone can just religate with itself. And if it religates with itself, then it will still transform properly, it will still confer antibiotic resistance, but the transformed colony won't have your insert, which is really, really bad. So restriction enzyme, digestion and ligation has to have even more screening going on. So traditionally, you would have laxed in or around the MCS, and then putting your insert in would disrupt the function of laxed, which means that on XGAL plates, XGAL turns blue when laxed metabolizes it. On XGAL plates, your unsuccessful transformants are blue. But this is an extra screening method after the orientation screening. So restriction, digestion, and ligation just has loads of issues. It's not used commonly anymore. It's not particularly good, and you probably won't be using it. But it is very simple to do. So preferably pick Golden Gate or Gibson, but this is technically an option.